Thanks for joining for the final session this afternoon. It's my pleasure to present Vesper Bio. And to give you a brief introduction into who we are and what we do, we're a private VC-backed biotech and experts in the sorting biology. We're now in clinic, entering phase 2. Sorting biology has been validated as a target of interest in a range of clinical indications, and we've defined our first program in frontotemporal dementia where sortilin is a clinically validated target because it interacts with a trophic factor called progranulin, which is important for neuronal health and survival. And so we're pushing forward with our small molecule antagonist VES-001, which is the first identified oral small molecule that interacts with the sortilin protein and crosses the blood-brain barrier. And in so doing, interrupts the interaction between sortilin and progranulin, which then can allow renormalization and redistribution of progranulin throughout the system and halting the horrendous disease of frontotemporal dementia. It's the VES-001 is the first of our clinical programs to get into phase 1. We will be developing additional programs for Parkinson's disease, retinal neuropathies, and a range of other indications that are amenable for sortilin therapy. So our first program, FTD-GRN, we're looking to push VES-001 forward on an accelerated approval pathway that will allow us to get to an NDA already in 2027. And to give you a flavor as to why that's possible, but also what we're trying to achieve, frontotemporal dementia, for those of you who don't know it, is a devastating disease. It's fatal. There are no current therapies, and it's one of the most common early onset dementias or degenerations, as AFTD would like us to call it. It's characterized by being early onset, I've just said that, by onset mostly before the age of 60, and death within 7 to 10 years of symptom debut. The first couple of years can be highly devastating for the behavior and personality of the patients. Whilst it's a rare indication, it is genetically defined, FTD-GRN. There are roughly 17,500 patients at any one time in the 7 major markets, but 140,000 carriers of this mutation at risk of getting the disease. And if you do have the pathogenic mutation, FTD-GRN, there is 100% penetrance, meaning that it's a death sentence for the individuals who carry this dominant gene. On this somewhat complicated slide, I'm hopefully going to talk you through the role of both sortilin inhibition and VES-001 in maintaining the health of microglia and neurons. So in the top we have, on the top right, we're displaying a microglial cell, and in purple below that, that's a neuron. And there's an important role of the I don't know if I can point of the progranulin, which is represented by a straight line of a number of balls attached to it. Progranulin is important for microglial health. It signals anti-inflammation to microglia. It's also highly important for neurons, where it is taken up on the back of prosapacin to mediate lysosomal function. What we do understand is that the sortilin is an elimination pathway for progranulin that in normal human beings or wild-type humans is important for maintaining the basal levels of progranulin. But in the case of FTD-GRN, where you have a haploinsufficiency or in the progranulin gene and a 50% reduction in extracellular progranulin, sortilin actually becomes a bad player. Now, sortilin and progranulin each have powerful biologies in their own right, And what we're doing is introducing Vesper-01 at exactly the node where these two powerful biologies interact, i.e. at the cell membrane. So the green pill called Vesper-01 is in fact a small molecule that is selectively inhibiting the interaction between sortilin and progranulin, therefore reducing the elimination rate of progranulin and renormalizing the distribution of progranulin, therefore, in the extracellular space, but also inside the neurons via the prosapsin-mediated pathway. So what we can demonstrate is that sortilin itself is a degradative pathway for progranulin. The appropriate pathway for uptake of progranulin to the lysosome is on the back of prosapsin as a heterodimer via the M6PR receptor. Our data show that we can indeed block sortilin-mediated uptake and shunt the uptake of progranulin over to the prosapsin-mediated pathway, thus renormalizing, redistributing progranulin throughout the whole system. And therefore, we anticipate halting disease progression. Very briefly on the candidate itself, it's a well-behaved small molecule. We designed it in-house, chiral, crystalline, etc., stable. We're currently in pivotal tox studies. It's a simple and efficient manufacturing process. Super importantly for the target indication, FTD-GRN, it gets into the brain very nicely. It has a nice oral half-life from human studies of 11 hours and is ideal for this indication that we're looking at. We have a focused team based here in Copenhagen, supported by a scientific advisory board made up of a number of well-known players to those who understand and know the FTD space. Clearly, this is an important aspect of driving forward an orphan disease program, getting involved with the patient groups, understanding where the key opinion leaders are, where the specialist sites are, understanding the natural history of FTD-GRN, and this is what our scientific advisory board helps us with. I'll note also Katie Brandt, she's a patient advocate, and just to give you a flavor of the devastating nature of this disease, her own husband died of FTD at the age of 33, just when their child was entering the second year of life. So this is a disease that can radically change people's lives and end people's lives tragically early. Wow, I'm quick today. So, just to come back to the very beginning, we are a clinical stage biotech in Vesper Bio. We are sortilin biology experts. We're developing a pipeline across various CNS and non-CNS indications with a range of assets that are unique to Vesper Bio. We have initiated phase 2 clinical development in FTD-GRN. That first patient, first visit will be in November of this year. We have a unique mechanistic differentiation to other players looking to treat these awful diseases. The FTD-GRN itself is being targeted with a clinically validated target in sortilin. The mechanism of action is understood and well characterized. Vesper-01 is the first small molecule that can drug this target, and we're ready to get into accelerated approval NDA in 2027. So that was brief. Any questions? Happy to answer them. Thank you.
Thanks for the nice presentation. Can you talk about the pipeline? Because my understanding is there are a few other assets, perhaps phase 3.
Yep.
Can you talk about the differentiation?
No, exactly. So Alector is currently in phase 3 with a monoclonal antibody. Monoclonal antibody requires a monthly infusion, around 60 milligrams per kilo or 5 grams of antibody once a month. So one level of differentiation is that we're oral and convenient home dosing versus the travel to a site for a multi-hour infusion, which given the type of patients and their own private lives is much higher impact. Now, the small molecule also opens up doors to go pre-symptomatic, which is also important. Now, a key differentiation, Alector is a degrader of sortilin biology, which may or may not have its own impacts. Our approach is a light touch on the sortilin biology that maintains all the beneficial effects of sortilin both inside and outside the cell. So there is differentiation. We mainly position it for the convenience of the oral home dosing, but there is also on a biological level. And the other pipeline right now would be 3 gene therapies, including, of course, Aviado Bio, and well done to them. They got a nice deal today with Astellas. And then there's Denali, who have been working on a progranulin replacement therapy with Takeda. I believe they're coming back to clinical development sometime soon.
And do you have any sort of phase 2 design, and how would that look like? Can you elaborate a little bit?
Yeah, so our phase 2 is going to be a 3-month study in the relevant patient population, looking at 2 dose levels before we move into a phase 2/3 seamless adaptive trial that we anticipate starting next year. The idea is to use that seamless adaptive trial to get the accelerated approval, but also have that confirmatory trial already fully enrolled, which I think is the exciting approach about it. And just to come back to the accelerated approval, the number of natural history studies that are out there, the Genfi study, the NSP, and others really do allow us to move down that line of accelerated approval because they have generated all the data that we could hope for. Yeah.
Thank you.
Thank you very much. Any further questions?
Thanks, everyone.